Good morning, ladies and gentlemen, and thank you for tuning in. The host of this morning's webinar is LipoTrue, and we are joined by Dr. Sylvia Pastor, who is Head of Scientific Research and Business Development at LipoTrue. She'll be presenting an overview of diving into your skin's microbiome to unlock anti-aging properties. But before we begin, I'd like to add there may not be time for Q&A, as it's just a teaser. But if you do have any questions regardless, please feel free to submit them through the chat feature, as we may have time for one or two questions. And please also leave feedback, as it will help improve our future webinars. So without further ado, I'll now pass you over to Sylvia.
Hi, everyone, and thank you for joining us on this presentation. LipoTrue is launching a new bacterial ferment with novel anti-aging activities, working on our skin microbiome. Its name is Ionome. This new active is produced by the fermentation of a bacillus strain collected from a protected subterranean cave in Majorca Island, which has been dated to be more than 3 million years old. The name Ionome makes a reference to the Latin name Ion, which means vital force and eternity, and also to the Greek god named Kronos. You can see the real image of the cave in blue on the right-hand side. When speaking about microbiome, there are three well-known concepts around it. Prebiotics, which are substrates for microorganisms. Probiotics, which are alive, helpful microorganisms. And postbiotics, which are metabolites released by microorganisms. With Ionome, we present a novel approach and a new concept, H-biotics, meaning a prebiotic for skin's microbiome, which enhances the release of H-less postbiotics. I will explain how now. On this slide, you can see a drawing of our skin and its main components, and with the shape of a stalactite, linking the origin of the active and the main applications on the skin. To evaluate the efficacy of Ionome, we have used an in vitro model of colonized skin using human epidermal keratinocytes and a combination of 5 main representative microbial strains. As a summary, here we are highlighting the following, which I will detail on the next slides. A positive modulation on the contribution of key microbial representatives on human keratinocytes, a direct antioxidant activity of Ionome under oxidative stress conditions, an increase of the ferritin antioxidant power, and the following characterization of the main contributing metabolites for this activity, which are released by the representative microbial strains through Ionome. A consequent improvement on the levels of proteins on human keratinocytes involved on integrity reinforcement, inflammation protection, and dermoepidermal junction nourishment. And a final clinical efficacy of Ionome on crow's-feet wrinkles and forehead, hydration, and a specific evaluation of skin microbiota of treated volunteers using next-generation sequencing tools. We start now with the in vitro efficacy evaluation. On this slide, you can see the direct protective effect of Ionome on human keratinocytes exposed to oxidative stress using hydrogen peroxide. On the left-hand side graph, Ionome increased the specific expression of catalase and ferritin genes on oxidative stress cells, hence stimulating the natural detoxifying mechanisms of keratinocytes. On the right-hand side graph, we demonstrate how Ionome is able to protect the keratinocytes from oxidative stress, decreasing the production of reactive oxygen species. We used ascorbic acid as a benchmark control, represented by the brown bar on the graph. Moving to the next slide, here you can see our in vitro model of colonized skin and how Ionome promotes the contribution of beneficial microorganisms. We used a specific ratio of 5 commensal skin microorganisms representing a healthy skin microbiome: Staphylococcus epidermidis, Staphylococcus hominis, Cucibacterium agnes, Corynebacterium cerosis, and Malassezia restricta. On the first bar of the graph, you can see the specific contribution of each of the microbial strains at the beginning of the treatment. On the second bar, you can see how, after 8 hours in culture, the microbial representatives decrease in numbers in the absence of any treatment, mainly epidermidis, hominis, and acnes, while Xerosis contribution gets more prominent. On the third bar, you can see how, after 8 hours of treatment with Ionome, there is a moderate and balanced increase of the main commensal microorganisms, more prominently of epidermidis and hominis. With these results, we can conclude that Ionome is having a probiotic activity on skin microbiome. We now check the biological activity that the treatment of Ionome exerts on the skin microbial representatives. On the top right front of the slide, you can see a summary of the interaction between Ionome and the microbial community and how the stimulated strains release specific antioxidant metabolites. We believe that this is a novel discovery. So for the first time and with Ionome, we prove how a novel bacterial ferment is able to increase the antioxidant power of skin microorganisms and to identify what specific metabolites or postbiotics are being released by them. First, we check the ferric reducing power of supernatants from in vitro cultures of colonized keratinocytes treated with Ionome after 8 hours. We can see on the left-hand side graph how Ionome increased FRAP by 7.57-fold, the yellow bar, when compared to untreated colonized keratinocytes, the blue bar. The benchmark quercetin was used as a positive control on this model, and Ionome shows a 2.7-fold higher activity than this benchmark. These same supernatants were later analyzed by metabolomics using liquid chromatography mass spectrometry for the specific identification of antioxidant metabolites involved on this FRAP antioxidant activity. On the right-hand side panels, you can see the 3 specific metabolites identified differentially on the condition of colonized keratinocytes treated with Ionome. Again, this is one of the big highlights of this product, since we prove the relevance of skin microorganisms on the protection of our skin health through antioxidant mechanisms. Coumaric acid is a very well-known antioxidant which has been described to be more potent than ascorbic acid or alpha-tocopherol. Tetrahydroisoquinoline is an antioxidant alkaloid described to be produced by certain plants and microorganisms. And aspartyl isoleucine is a dipeptide with known antioxidant activities. On this slide now, we complete the final picture of the mechanism of action of Ionome by studying its effect through the microbiome on epidermal keratinocytes using proteomics. Using the same in vitro model of colonized skin, the treatment with Ionome for 20 hours reported the upregulation of key anti-aging proteins by primary keratinocytes. On the graph, we are showing the fold induction increase of the different proteins found. On the left, you can see the drawing of the cytoplasm of a cell, showing the location of the newest proteins identified and their relevance on key processes of structural integrity. Related to cellular cytoskeleton reinforcement, we found radixin and moesin as part of the ERM protein complex, wave 2, filamin, and vinculin. All of them interact with the actin filaments of the cells. And also we found tubulins. We also observe an increase on the levels of proteins involved in the dermoepidermal junctions, such as keratin 79 and collagen 7, and 2 additional proteins, one with antioxidant efficacy, SOD1, and one protecting against inflammation, annexin 1. We move now to the clinical testing of Ionome. Briefly, we used two panels of women with ages between 40 to 55 years old, all of them with a healthy skin. One group was treated with a placebo cream, and the other group was treated with a cream containing 3% Ionum. The products were applied on the whole face twice a day for 28 days. Before and after 28 days of treatment, a cotton swab was applied on the forehead of each volunteer to get a sample of their skin surface microbiome for next generation sequencing evaluation. On the graph, you can see the effect of Ionum after 7 days of treatment on crow's feet, the yellow bars, and placebo in blue. There was a significant average decrease of 8% in the area of the wrinkles and a 17% decrease of volume after the treatment. On the right, you can see a representative 3D image of a 50-year-old volunteer treated with Ionum. On the second slide of the clinical evaluation, you can see the effect of Ionum on the forehead of the volunteers after 28 days of treatment. On the graph, you can see a significant decrease of the average wrinkles area of the forehead by 10%. And on the right, you can see real images of the before and after of a 38 and a 43-year-old volunteers. Ionum also showed an improvement on skin moisturization after 1 week and 1 month of treatment, with significant increase in values of 8% and 9% respectively. This last slide highlights the effect of Ionum on skin microbiome and links our in vitro studies with the clinical results. As I mentioned, for each volunteer, a swab sample of the skin's microbiome surface of the forehead was taken before and after 28 days of treatment. All recruited volunteers were not showing any type of dysbiosis, as this new active is meant for its use on broad-spectrum skin types. We used next-generation sequencing analysis to identify the operational phylogenetic units of more than 7,000 microbial species present on the skin. The main results obtained showed that the relative abundance of all these species were not altered by Ionum, in consonance with healthy skin microbiome, which is able to balance naturally. As a plus, Ionum showed a modulation of the relative abundances of key relevant microorganisms as observed on the in vitro model used. There was an upregulation of S. hominis and Corynebacterium species, important on moisture areas and described to release antioxidants. There was finally a trend by Ionum to maintain the contribution of Staphylococcus epidermidis, while placebo showed a slight decrease, and there was also a decreased modulation of C. acnes, slightly increased by placebo. As a result of this balance and modulation of key species, Ionum links nicely its in vitro activities with the clinical results obtained. This wrap-up slide summarizes in a very visual way how Ionum, produced by a bacterial strain, collected on a protected subterranean ancient cave, is capable of unlocking, you can see from left to right, the natural processes in which our skin microbiome, through the release of antioxidant metabolites, balances, nourishes, protects, and takes care of our skin to rejuvenate and improve its quality. This is the technical summary of the product. Ionum is a biotechnological ingredient, compliant, Cosmos approved, and 100% natural according to ISO 16128. And it's also biodegradable, preservative-free, and water-soluble. Hope we have managed to catch enough interest from your side. In LipoTrue, we are aware of the current global situation due to sanitary emergency. But still, all our sales team and distributors are fully available on your regions to talk to you more by teleconference and to present to you our full version of the product and answer any questions. Also, we will be having a more extended seminar on April 23rd. Stay safe and thank you for your attention.
That's great. Yeah, and that was all about our webinar for today. And I'd like to thank Sylvia and LipoTrue for hosting. Sylvia will also be presenting in a technical seminar at In Cosmetics Global, and we hope to see you all there. This webinar will be available to view again on demand, so do feel free to visit the channel again. And full details of the rest of today's webinars are on the website, so please do check those out. And with that, thank you so much for tuning in, and take care.